I am going to talk about the language of diseases, and this is just a fancy way for me to kind of say this is just one big vocabulary test. Um, and it might sound kind of, unimportant, but I think it's really important that we understand these words that we're going to come across when they referring to disease and just have an understanding of how they're used and what people mean when they are using them. All right, so here we have some pictures. Can you see my mouse? Yes. Okay. Or maybe not. Um,
I don't see your mouse.
you can see it circling the, squash. Can you see it now?
Yes, now I
Oh,
can.
okay. All right. Here we just have a squash with a virus. And then here are just some tiny little baby stink bugs hatched out. All right, so let's begin. Maybe. Okay. So the objectives today, we really want to learn why it's important for us to, understand some of the vocabulary, the basic vocabulary. Um, we're going to go into signs and symptoms, which is a really common term that you learn when you're first learning about diseases. Um, and then we'll also understand a lot of vocabulary terms, so it's a big vocab quiz today. And then we'll have a bunch of practice, a little bit of practice throughout, and then at the end we have some practice again. I will also say I'm a little bit under the weather today, so I apologize if you can kind of hear it in my voice. And I just will send a tiny prayer up to the mycelium network that I don't have a coughing fit at some points, but we should be good. I'm a little bit better today. All right, so why are we going to learn disease? The most important thing is we just want to have clear communication, and this is in regards to everything we do. We'll learn some terms, and it's always important to use some of these more technical terms, but a lot of times just using more descriptive terms, whether they're technical or not, will be beneficial. So we just want to make sure we know that we need to communicate clearly when we're discussing problems with someone else. And this is important with diseases because a lot of times, unless it's a very characteristic diagnostic disease, a lot of times you need help from some other people. So experts, you send pictures and emails, or you can send a sample off to a diagnostic lab, but you're going to be communicating these problems with someone else. And if we understand how to identify disease and be able to communicate them, we can hopefully catch them a little bit earlier on. We also want to strengthen collaboration between homeowners and the master gardeners, and then master gardeners with researchers and extension agents and all of that. And one thing that researchers aren't always very good at doing is being able to communicate their expertise in a way that most people can understand. So in this way, we kind of want to bring everyone up to the middle. We want researchers to kind of be able to simplify their ideas, but when you were trained master gardeners, we also want to be able to kind of understand some of the more complex terminology. All right, and then the last thing is we would never expect anyone to become an expert in diseases. I was studying diseases for many years, getting my doctorate degree, and I worked in a diagnostic lab, and my favorite thing about it was how every single day I was stumped and confused. It adds some excitement, but it's like no matter how confident you feel you get in diseases, something will always come along that just completely stumps you. So even the experts have problems, so we're not here to become experts, we just want to get a little bit more knowledgeable. All right, so here's just a really quick example of something where if I had a picture of this, and someone said something's wrong with my plant, if I were to send this to someone, it would be really hard for them to be able to figure out what's wrong. And maybe in the future we can do a bigger webinar series about how to get good pictures and send in good diagnostic photos and good ways to send. We can even discuss the digital diagnostic network a little bit more. But something like this, if you send that to someone, they may not know what's going on. Versus something like this, where you say my Arcadia, which is a variety of blueberry, is in 20-gallon pots. They're experiencing branch dieback and chlorotic stems. This may not mean much to you, but if I see this and I see this, I can actually kind of tell what's going on. So I know this looks a lot like algal stem blotch. Here with the white background, I can see it's just one branch that's dying off. That's a lot like Botrosphaeria dieback. Here's some galling of the roots. So again, you might not see much here, but when you send a really good, clear picture to an expert, they can usually figure out what's going on or at least get a bigger clue. So even if you're not using technical terms, just be as descriptive as possible and the best photos as possible. All right, so before we begin, we really just have to define disease, and this is going to be the most technical slide because, I mean, you guess you just want to lay it all out there. So the exact definition of disease is the series of invisible and visible responses of plant cells and tissues to a pathogenic organism or environmental factor. It results in an adverse changes in the form, function, or integrity of the plant and can lead to partial impairment or death of plant parts or the entire plant. And a little fun fact about Agrios, who wrote the book on plant diseases, pretty much. He is a University of Florida professor. He was the department head of plant pathology for a long time, but he also founded the Doctor of Plant Medicine degree program. So he founded my degree that I eventually graduated from. So fun fact, Dr. Agrios is kind of a superstar in my world. But this is disease, and so we'll split it up into a little bit of a little sections. So first we have physiological malfunction. Basically, this means disease is going to disrupt the cellular chemistry, the cellular processes of the plant. As opposed to insects that might just consume part of the plant, diseases have a really intimate relationship with the plant. They are disrupting some of the actual biological processes. So we have some disruption of that. Diseases are a continuous state, and this is what separates disease from damage. And there is some gray area in here. And as you'll hear me say a lot, there's gray area everywhere. So some of these words, it's impossible to put any form of biology in a box. Diseases are just like that. So in general, when you have a disease, it's something that is prolonged or a continuous state. So a continuous infection by a fungi. Disease can also be a continuous nutrient deficiency due to some environmental factor. Damage is a one-time event. So this is something that happens usually really quickly. So a freeze or a drought. But of course we get or insect chewing or damage like that. There's that middle ground where like, well, what if the drought lasts, a couple days? That's pretty continuous. So is that disease or damage? So there is some gray area. I will also say that I learned growing through my degree that disease is more relying to a pathogenic agent. So it's more biotic in nature. So biotic is living. And then disorder is more abiotic or environmental in nature. So like a nutrient deficiency is technically, through this definition, a disease. It's an environmental factor. But some people would call that a disorder. And in general, I kind of use disorder for environmental factors, disease for biotic or living factors. So you'll hear both of those. And then finally, a disease is caused by, again, either a pathogenic agent, which is fungi, bacteria, and viruses primarily, or environmental factors like stress or pollution. But again, a lot of times with environmental factors, we will consider that a disorder rather than a disease, but you'll hear both sometimes. All right, and then finally, you'll see some symptoms. So again, this is related to how it disrupts the actual physiological processes of the plant. All right, so let's briefly go into pathogen because there's not entirely a consensus on what a pathogen is. In general, it's an evolving entity that causes plant disease. And so things that are definitely pathogens are fungi, bacteria, viruses. In addition, phytoplasmas, which are a type of bacteria. We'll get into that. And then oomycetes, which are a type of fungi, which we'll also get into. So these ones are definitely pathogens. Everyone agrees that they're pathogens. They disrupt the, cellular biology of the plant. They cause disease. Some things that also are pathogens are nematodes. So nematodes are non-segmented roundworms. So they're microscopic roundworms. They live in the soil. They're everywhere. We could do another presentation on nematodes if we ever wanted to. Really interesting. Nematodes are sometimes grouped as pathogens, sometimes grouped as pests, kind of like insects. They do sometimes cause like gallings on roots. They do sometimes cause diseases. So sometimes they are pathogens, sometimes they're pests. And you can see this with the layout of a lot of universities. So the University of Florida, we have the entomology and nematology department. So bugs and nematodes are in one department. In other universities, plant pathology and nematology are grouped together. So the plant pathogens and the nematodes are grouped together. So nematodes are just this, kind of outside group. But insects, mites, slugs, other pests, these are pests, not pathogens. They tend to not cause the disease, so they don't really disrupt the physiological function of the plant. However, there are some insects and mites that do kind of have an interaction with the plant. So again, that makes it a little confusing. But in general, fungi, bacteria, and viruses, pathogens, everything else, pests. All right. So now we're going to get into the first thing that you really need to learn when it comes to diseases, and you might have already seen this before, it's the disease triangle. And this is simple concept, but it's really, really important that we understand that disease, you really need to have all three conditions. So you need to have a susceptible host, a virulent pathogen, and a favorable environment. Many people think that if you have a host present and there's a pathogen flying around in that area, you will have disease. But that's not the case. We really also need environmental conditions that favor the disease progression. So that's important. It's also important because, as we know, what we have the most control over is the environment. So we can control irrigation, which controls leaf wetness. We can control spacing of plants, which controls the humidity and aeration. So we can control a lot of the environmental factors. So we can control some disease. So yes, we need all three of these to be occurring in order to get disease. Anything that we're missing any of these conditions, there's no disease. All right, so here is one of the first things you will ever learn if you ever take a plant pathology course, and it's the difference between sign and symptoms. And so in general, the basic definition is signs are really referring to the pathogen, and symptoms are referring to the generalized plant. So with signs, it's you physically can see some evidence of the pathogen. So this is usually fungi spores, fungi hyphae or mycelium, and bacterial ooze is also a sign. So in this picture this is a blueberry stem. It's inside a graduated cylinder, and this white stuff that's just kind of oozing out of it, this is actually the bacteria. So this is just giant masses of the bacteria oozing out of the stem. So this would be a sign because we can actually see the bacteria. Obviously, we can't see individual bacteria, but like we know in here, that's all bacteria with some of their, exudates and stuff like that. And then for the strawberry, we have all this white, fluffy, kind of gray, fluffy stuff. That's hyphae, mycelium. So that's actually the body of the fungus. And then this little orange stuff, that's actually just masses of spores being produced. And so again, we can't see any individual spores or the individual hyphae, but this is all the actual fungus. So that's a sign. And signs tend to be very specific. So if you see a sign, you can pretty much tell what the disease is or what pathogen it is because it's related to the pathogen. With symptoms, they are more generalized, and this is a plant response to some pathogen. But symptoms can also be responses to other things other than pathogens. So environmental stresses, damage, or anything else. The plant just has a few symptoms that it's go-to when something is wrong. So we have stuff like wilting, yellowing, and stunting. So this is palm that's showing wilting and chlorotic. And then there's some chlorotic leaves. Again, we would have no idea what's going on unless we knew any more about the situation because the plant symptoms can be for a variety of different things. They will have the same symptom. But in summary, symptoms are the plant's response. Signs are actual indications of the pathogen. All right, so we're going to go through some of these later on, but just some common plant symptoms are wilting, chlorosis, and we have chlorosis. This could be for anything, so it can be nutrient deficiencies. It can be a pathogen related. So we don't know what's causing these symptoms, but they are a symptom that the plant is displaying. Necrosis, again, this can be cold damage. It can be a pathogen. It could be normal leaf senescence if it loses its leaves in the winter. We don't know the cause, but this is just a generalized plant symptom. Distortion. So this is anything kind of funky. We like to say is distortion. A lot of time viruses, and in this case, it's a phytoplasma. We'll discuss a little bit later. Um, Distortion can be a number of different causes. It can also be herbicide injury. So again, we don't know the cause, but we know the leaves are kind of funky. They're distorted. Stunting. Again, this can be a nutrient issue. In this case, they're stunted because of nematodes in the roots. So these ones have high volumes of nematodes. These ones don't. These ones are stunted. But it could be something else. All right, so common pathogen signs. So here we have fungal mycelium. So all this white fungal growth, that's the actual fungus. So we can see the fungus, it's a sign. Here is again bacterial oozing or bacterial streaming. That gooey, goopy stuff is the actual bacteria, along with some of its, carbohydrate-rich exudates that kind of goops them together. And then as I discussed, insects are not pathogens, but when we're referring to insect pests, the insect itself is going to be the sign. That's just one way to think about it. The actual insect is the sign. So like the fungus is the sign or the bacteria is the sign. So the actual pathogen or pest itself. All right, so here we have a little quiz. So this should be hopefully not too hard. This is a different example of something I already showed. It's a blueberry stem. And what I'm specifically referring to is all this white stuff that's streaming down this water. Now, do you guys think this is a sign or a symptom? And you can speak or in the chat maybe, or I'll just give you a couple seconds to think about it, and then I will go into the next slide. All right, someone said sign. More signs. Okay, you are correct. It is a sign. So this again, this white stuff is the actual bacteria. This is Ralstonia solanacearum. It's a blueberry pathogen. It's actually can be a regulated pathogen in some areas, so it's not great. We do have an endemic version. But again, that white stuff is the actual bacteria. One more example. Is this a sign or a symptom? You guys are so smart. This is a symptom. So this is bacterial blight on geranium. It's caused by Xanthomonas campestris pathovar pelargoniae. So basically, we can't see the bacteria anywhere on here. And the only way we know it's caused by a bacteria is because we did the testing. But right now we can see dieback, you can see wilting, but you can't actually see a bacteria. And as just a little fun fact, for a lot of pathogens, species is just not specific enough. So a lot of these will have even more specific names. And so in this case, it's pathovar, PV, pelargoniae. So that's a more specific genetic group within that one species. And this one only infects geranium. So yeah, sometimes species is just not enough. All right, so now we're gonna move on. Actually, I'll ask, are there any questions? All right, I don't see any. So the next thing that we're going to talk about, and these are terms that you may not actually hear a lot, the actual terms, but I think they're really important to learn about and to understand because these terms really accurately describe how pathogens live. So what they are doing, how some of them get their food source. So some pathogens do it a little differently than others. And so we want to kind of understand the different ways that some pathogens can live. So what we group these in, and these are a continuum. It's definitely not a strict grouping. We have biotrophs, and then we have necrotrophs. So these are the opposite ends of the spectrum. And if you just think about it, like think about, the terms bio is living, troph is like on the food chain. And then necrotroph is dead or necrotic tissue. So it has something to do with living and dead tissue, kind of. You can kind of guess that. And in the middle, we have semi-biotroph, sometimes also hemibiotrophs, so you can use both terms. So I'll go into them, but, kind of keep that in mind. So biotrophs, so these are the living eaters. They are or pathogens that can only feed on living tissue, living cells, living plants. So they need a living host to grow and reproduce. They do not compete well in the external environment. And what we mean by this is if you have a plant that is killed off by a pathogen and you have just the leaf litter or the dead plant in the soil, everything else that's in that soil, there's a lot of fungi, a lot of bacteria in that soil, it will all outcompete it. It will not compete well once the plant dies. So it only can compete if it's a living plant. And an example of this is over here we have downy mildew on lavender. So if you see all this gray fuzzy stuff, that's actually the downy mildew. So it's literally everywhere. And you see the tissue is still pretty green and living because downy mildew is a biotroph and needs living tissue. If you have something right here, if I were to culture this out, um, I would not find any downy mildew on there for sure. I would find something else because once this tissue starts to die, then everything else starts to outcompete the biotroph. So they compete well only with living tissue. Most cannot be cultured in isolation. This is a problem for scientists everywhere. We like to have evidence that what pathogen we isolate, we can put it into a healthy plant, and that means we 100% know that this is what's causing the disease. But when you have biotrophs, you cannot culture them, and how we culture them is it's typically in just like little petri plates, which is just potato jelly, pretty much. So if you know about agar, you have just potato sugar and agar, and that will grow out fungi, some bacteria. So we like to grow them out there. That way we grow them outside of the plant to kind of remove all the confounding factors. But in this case, we can't usually do that. So it adds some complexity for scientists. Obligate parasitites, which means they can't exist in the environment. They have to be parasitic on whatever host they have. So one of the most common examples is rust. These are two examples of rust. And rust are really, really fascinating and really, really complicated because not only do they need a living host, but a lot of rust need two living hosts. So they will have two different phases, one on one host and one on another host. So it gets really, really complicated. And then downy mildew, powdery mildew is another example of biotrophs. All right, so now we're going to go to the opposite end of the spectrum. So biotrophs need living tissue in order to survive. Necrotrophs don't. They actually need the tissue to be dead. They only consume dead tissue. So what necrotrophs do is they consume dead tissue, obviously. They can colonize living tissue, but they have to kill the tissue before they consume it. So they will colonize living tissue, but then they will kill it and consume it. And a lot of times they produce toxins, which is what, ends up killing the living tissue so that they can slurp up all the juices. And they are facultative parasites and opportunistic pathogens, which basically means they don't have to be pathogenic. And a lot of times this is in the case when environmental factors are mostly into play. So when the plant gets stressed out, they tip over to being a pathogen. An example of this is take all root rot. This is really, really, really common in St. Augustine lawns. If you send me a sample of a St. Augustine lawn in the summer, I guarantee you I will be able to find the fungi that causes take all root rot. However, it does not usually progress to this stage unless there's some environmental condition. A lot of times this is compaction or drought or some other thing. So they are more opportunistic. And then this is southern blight, Altheliovulfia in watermelon. You can see this is the actual fungal mycelium. So this is the body of the fungus. You can see it started here and it's just killing its way over this watermelon and slurping up all the juices as it goes. All right, so those are necrotrophs. Now we're going to go through everything in between. And again, most things find themselves somewhere in between. And these are semi-biotrophs or hemi-biotrophs. Both words are used. And so these ones will typically have two different stages. So a lot of times they will initiate infection and feed on living tissue for a time. Sometimes this is really short, sometimes this is the majority of the disease. But for some phase, they have a biotrophic phase. And then at some point in time, for some reason, maybe it's some trigger that we don't always understand, they will switch over to a necrotrophic phase. So this is when they start killing off the tissue. The disease tends to progress more rapidly at this point. Um, and this is also when they might be able to survive in leaf litter. So sometimes you have these semi or hemibiotrophs can be big problems because they can survive in the soil or in leaf litter or in other hosts for periods of time. With biotrophs, you tend to, if you can control the host, and it's not like some rust have, their spores can go in the air for miles and miles, over states, in fact. So for some of them, it's hard to control them, but if you control the host, get rid of all the host, they can't survive. But with necrotrophs and hemibiotrophs, they can sometimes still survive in the soil if it's soil borne. All right, so that's basically the concept of how different fungi live. So some of them really need living tissue, some of them need more dead tissue. So that's just categorizing the different ways, and it's not fungi, it's also bacteria. I think mostly in fungi terms usually. Um, okay, but now I have another question. And so this one is, there's one group of pathogen that is only biotrophic. So you just have to do a little thinking. So which group of these, fungi, bacteria, nematodes, or viruses, which group can only survive on living tissue? So most of these groups have a combination of the biotrophs, hemibiotrophs, and necrotrophs. All right, two people have said viruses. Well, if you said viruses, you are absolutely correct. Viruses they need to take over the cell machinery of the host in order to reproduce more viruses. So if a dead cell cannot be hijacked into creating more viruses. So yeah, all viruses are biotrophs. So just a little fun fact. And then here's just an example of a virus particle with an electron microscope. You cannot see these with your naked eye. You definitely can't see them with a regular microscope. And then here's some cool symptoms of tomato spotted wilt virus. Some viruses just have really, really ornate and cool symptoms. All right. So now we're going to switch over to more pathogen related terms. So these are going to be terms that are typically referred to by fungi, typically bacteria related or virus related. Do we have any questions before now? Okie dokie. If not, then I will continue and you can just put it in the chat. All right, so we're going to start with fungal terms. I've probably already used this term a couple times, so sorry. But let's just refer to what hyphae is. And again, you probably know what hyphae is, but we just want to make sure we have it down. So hyphae is the threads, the filamentous structure that makes up the body, which is the mycelium of the fungus. So this is what is actually the fungus. When we think about mushrooms, that's the reproductive structure of the fungus. That's a tiny, tiny fraction of what is actually going on with this fungus. And the hyphae is what is actually causing the fungus to grow. It is what's taking in nutrients. So the way that fungi eat, they are heterotrophs, so they don't photosynthesize. They have to get their nutrients from the environment like the rest of us. And so the way they do it is they kind of just digest their environment. So they excrete digestive enzymes. Those go into, their environment, they kind of soup up their environment, and then they can absorb those particles. So that's how they consume. They just keep growing and keep, melting away their environment and eating it up. And so here is hyphae under a microscope. This is on a rhizoctonia. And so this is actually a diagnostic growth habit of hyphae. So if you see a diagnostician, we can definitely tell what type of fungus it is, judging by the way the hyphae is growing. So sometimes hyphae can be really diagnostic. But most of the time, when it comes to hyphae, you need to see it under a scope to see the individual hyphae, which is why we get into mycelium. Mycelium is just the body. Really, it's just, if you can see it with your naked eye, it's probably mycelium, not hyphae. It's just, we like to have a term for everything. So in this case, this is just the body of fungus. It's just a bunch of hyphae together. And so one way to think about this is if you have like a blanket or something, each individual thread is the hyphae, and then the mycelium is the actual fabric, so the blanket itself. And here's just an example of a bunch of, hyphae on a plate that's, you can see with your naked eye, so it's mycelium. And then I also have a little fun fact on here. So this is a point that's sometimes contested, but the largest living organism on Earth is actually a honey fungus, which is Armillaria. And it's a single specimen is in Oregon's National Forest, and it spans 2,240 acres, which is about 3.5 square miles. So it's really, really extensive, but it's mostly underground, and so that's why it's kind of contested. It's estimated to be 2,000 to 8,000 years old. Um, and so it's really, really old. And the reason why we can tell that this is one single specimen is with genetic analysis. So, you take a sample, then you go, three miles that way, and you can take another sample and they're genetically compatible. So it's just this one fungus that keeps growing and it's basically consuming a whole forest. And it's degrading the down trees and everything. So it is technically the largest organism that we know about. But of course, some people don't like this because you can't really see it. All you can see is occasional mushrooms, and these mushrooms pop up randomly. So some people would say it's, redwood tree is the biggest organism, but I strongly believe that it is a fungus. All right. So another fungal term that you will hear a lot is a spore. So again, we probably know what a spore is, but just in case, it's just the reproductive unit of the fungus. Sometimes we can also refer to it as inoculum. So inoculum is like the spores that will cause the disease, that will start in an infection. And then on here, I have two examples of different spores to kind of show how spores can look very different. And this is just like two ends of the spectrum. Spores look like anything. This is a really good way that if you um, are trying to diagnose a problem, like a lot of times we use the spores for identification. But here's Corynespora, which is a type of fungi. And you can see right here, that's a spore. That's a spore. They kind of grow in chains. This is a spore. And I don't know about you, but if I didn't know what I was looking at, they look kind of like hyphae. And that's a distinguishing feature with this type of fungus. Their spores kind of look like hyphae. They almost start off as hyphae and they separate each other out. So sometimes spores look kind of like hyphae, so it's hard to tell the difference between the hyphae and the spores. And then here is a zoomed in picture of a leaf spot on a blueberry leaf. And you see all these little black structures, which by the way, have their own name. They're called pycnidia. So these structures all have massive amounts of spores just being produced, and they exude out of them into these little like noodle type, almost looks like ingrown hair in a way. And so this is just masses of thousands of spores. And then when you put them on a tape mount, you slide them into a microscope, this is what the spores look like. So again, very different from these spores. All spores have different structures, really important with identifying fungi, not always what you need because some spores look the same. A lot of times, we fall back on molecular techniques these days because it's really hard to differentiate some spores. But yeah, so here's spores, lots of different structures. All right, mildew. So this is something you will hear referred to by some, and mildew is technically just whitish fungal growth on plant surfaces. So there's two common diseases that will commonly be referred to as mildew. You have downy mildew, which I already showed you, and this one is a little confusing because sometimes it's more gray, and this is an extreme example where it's very gray looking. So it's whitish fungal growth, but like sometimes it's a little grayer. Then we have powdery mildew. Powdery mildew is one of the few diseases that are pretty easy for most people to recognize, which I always love. It's always nice having something that's most people can identify. But again, just whitish growth on the plant surface. All right, sclerotia. This is a term you may not have heard of before because not all fungal pathogens create sclerotia, and if they do, they're not usually very noticeable. But sclerotia is just a hardened fungal structure that survives tough conditions. So these are kind of the enemy. They can survive in the soil for decades even. So if you have a very aggressive pathogen and it creates sclerotia, that can be a problem. And basically it's just hyphae. It's massive hyphae that's just really tightly balled up, and it's just the outside is hardened and it can just survive any conditions. And somehow it knows when the host is, replanted back in the soil, and it will germinate. So this is an example of sclerotia, southern blight on watermelon. If you remember, I think it was like the first picture I showed of the or it's of the necrotroph where the watermelon was just being like consumed from one side to the other. That's the same pathogen. There's little orange little seeds in here. They look just like kind of seeds, and they're the sclerotia. And once the watermelon completely dies off and it becomes part of the soil, those sclerotia will stay and they will persist. Here's another example on a petri plate. Again, this is made up of just potato, sugar, jello, pretty much, that the fungi really love. So in this case, and we have like whole cookbooks related to how to grow fungi. Some fungi prefer specific concentrations, and then we have different things for bacteria. So it can be really cool. But all these little specks are also sclerotia. All right, so now we have anthracnose, and anthracnose can sometimes be a bit confusing for some people, and it's because it's referred to sometimes as a symptom, sometimes as a disease, and sometimes as a pathogen itself. So the symptom anthracnose is black sunken lesions on leaves, stems, or fruit. So typically the necrotic lesions are very black, typically necrotic spots are brown. These ones are brown to almost black. They tend to be sunken, and they can be on pretty much any plant parts, above ground plant parts. So where there's some nuance is, again, we have the symptom. So this is a symptom of anthracnose, but also a lot of diseases are named anthracnose. So this disease, the symptom is anthracnose, but also the name itself is also anthracnose on cucurbits. So if you look into a book and it says this is disease anthracnose, it's a disease, but also a symptom. And then it gets even more confusing because there's one specific genus of fungi, they're called Colletotricum, that most of the time causes anthracnose symptoms. And so sometimes, and I came across this a lot when I was working in a lab, where they would say, oh, there's anthracnose on this plate. And I'm like, that's Colletotricum. So we're referring to the actual fungi, it's Colletotricum. But because this fungi causes anthracnose symptoms so often, sometimes it's referred to as anthracnose, but it's not the only fungi that can cause anthracnose symptoms. But it's just a word to be familiar with, have it on your periphery, anthracnose. All right, so now we have target spots. So this is very fungi. So if you see target spots, almost always you can think, that's probably a fungal spot. And so it's concentric rings forming within the lesion. And this is because fungi grow radially. So they'll start at one point and then they grow in almost a perfect circle, if given the opportunity. Obviously, sometimes things hinder them. So in this case, whatever this vein was, it was just too hard for the fungi to cross. But in general, fungi will grow and they will just grow radially, and you can see these kind of target spot circles. And then this is an example on fruit. And this is another one where this is a target spot symptom. A lot of names are named target spot. So this disease is actually called target spot on tomato. So if you Google it, that's the disease name as well as the symptom that it shows. All right, and then we're also going to go to shot hole. So this is another thing that you will hear, and this is when you have fungi lesions that will continue to grow. And so they get kind of big. And then that necrotic center just kind of gets crispy and it like falls out. And so it looks kind of like someone took a BB gun and just put a shot hole through that necrotic spot. And so that's why it's called a shot hole symptom. And so this one is actually also target spot. So it's also a target spot symptom. Kind of hard to see with this picture, but you can really see it has some of those concentric circles. And a lot of times target spot symptoms, as they get larger, will eventually have that shot hole appearance. Is lethal browning caused by sclerotia? No, it is not, but we will get into what causes lethal browning or lethal bronzing, I think. So this is caused by an insect, so I just want to include that, where most shot hole are fungi related. It's usually associated with fungi, but this is caused by a leaf miner, so it's not always related to a fungi. And if I go back here, you can kind of see on this fruit, you almost, even though it's a fruit, not a leaf, you can kind of see a shot hole appearance almost starting. All right, so now let's just add something in here that I think it's important to note if you're ever doing any kind of research on any disease and you come across there's a fungi that you're researching and has two names, and why is this? And a lot of times you'll see the name and then in parentheses a different name. And you're like, what? Sometimes it has three names. But most fungi have two names. And the reason for this is most fungi have two different life cycles. They have a sexual life cycle and an asexual life cycle. So a sexual life cycle, they will combine genetic material, sometimes with themselves, sometimes with different, versions. They don't really have males or females, but they can combine genetic material. And so that's the sexual stage. And they have an asexual stage, which just, clones itself, reproduces that way. The problem is these two stages look very, very, very different. And so when we were identifying fungi based on morphological features, it's pretty understandable that a lot of scientists named them different things because they assumed they were different fungi. And then now that we are in our molecular age, we can do molecular tests and we realize, hey, these are actually the same fungi, just different stages, with different life stages of them. So a lot of fungi have two names that way. There is a effort to try and standardize this by having all of their names be the sexual version. So, standardizing it, so we just call them all the sexual version. The problem with that is the asexual stage is usually more common, and it's the one that we are more well known of. So it's hard to get people to stop using the more common name for a name that is less common. And then something else you may hear about is imperfect fungi. This is not fungi that are not good enough. This is just a not evolutionary group. So it's just a grouping random. It has no basis on, the genetics or evolution. Of fungi that have no known sexual stage. So we only know about their asexual stage. So maybe they lost their sexual stage, or often their sexual stage has just never been discovered. It's very cryptic. It's hard to discover fungi. So those are called imperfect fungi. They only have an asexual stage. And then here's an example of something I came across when I was in the lab, and I just think this is really cool. So these two this is the sexual stage of a fungi. Now they're different species, most likely. I didn't, get them down to species. I only keyed it out to the genera. So they're probably different species because they're on different plants. So this is on podocarpus, and this is on peanuts. They are the same exact genus of fungi. This is sexual. So if you can see, this is a root. So there's tiny little balls just on those roots. And if you put them on a microscope and you smush them, there's these sacs that are filled with spores. So that's what it looks like. And then you have this, which is on the roots of podocarpus. It's a brownish hyphae. And then you look at here and it has cylindrical spores, which is kind of why it's called cylindrocladium. And so that's what the asexual stage looks like. So as you can see, it's pretty understandable why someone would look at both of these and be like, that's different fungi. But they're actually the same fungi, just different stages, different species probably. So the more you know, this is why some fungi have two names. So don't be confused when you see two, if not more names. Fungi naming is just so confusing and it's always changing. So you have that to look forward to. All right, next thing we should do before we move on from fungi is we really need to discuss what on earth is an oomycete. Again, you probably maybe have heard about oomycetes. They are not true fungi. So they are water molds, aka they are closer related to algae. Fungi are kind of related to insects. I mean, not related, but they're closer to insects than they are to oomycetes. So this is an example of convergent evolution. So basically they're vastly different in the whole web of life. They're very different, but they've converged to have very similar life cycles or lifestyles, and they look very similar to the untrained eye. So here is a true fungi. Here is an oomycete. This is many people. They look very similar. It would be hard to tell the difference. It takes a trained eye to be able to tell the difference. But this is important for everyone to know because oomycetes don't always respond to fungicides the way that true fungi respond. So sometimes if you treat something with a fungicide and it's not working, it's because it's an oomycete. So it is not a true fungus, even though it looks like a fungus. And here's a little table. And I did send a PDF version of this presentation to Kathy, and so she can send it out to everyone. So if you want to come back and look at it, this is just how fungi and oomycetes differ. I think one of the important things to highlight is oomycetes, their cell wall is made out of cellulose, which is like plants, and again, they're closer related to algae. True fungi have a cell wall that is composed of chitin, which is the same material that insect exoskeletons are made out of. So we can see this is one example of how they are very different. And there are some fungicides that target the cell wall of the fungus, but if it's targeting the chitin, it's not going to affect the cellulose. So some fungicides are very broad spectrum and they might do some work with oomycetes, but it's never given. So you want to make sure if it's an oomycete, we get that identified. And it's hard to do that unless you send it off to a lab for someone to actually culture it out and diagnose it. All right, so I think that is it with fungi. Fungi is the longest section because it's complicated and it gets more complicated there. But are there any questions so far? All right, so I don't see questions. We can always answer them at the end. So now let's go into bacteria. So when it comes to bacteria, there's one symptom that is almost universally any way to get rid of oomycetes. We can have a different presentation that is more on like treatments. I think that could be a good idea. But one thing I would say with oomycetes is they really love water. So they have this thing called zoospores, which I think I mentioned it. So they have motile zoospores. So their spores can actually move, which is really fun. I had this video and I couldn't find it for some reason. But this is also a fun thing to kind of do with kids is you inoculate something with an oomycete and then you put it in water and then within a couple hours you see all the spores swimming around. So it's just kind of fun because they actually move as opposed to fungi. You can't really see them move. But they really, really love water. So a lot of times if you control the water, you control the oomycete. But there are also fungicides that target oomycetes, not regular fungus. All right, so back to bacteria. So one of the main symptoms referring to bacteria is water soaking. So if you have a leaf that is infected with bacteria, and again, bacteria also very water related, they kind of go where the water is going. And so when you look at this leaf, so like let's look at this leaf and for example, if it was on the ground, and this is actually where I would, when I was working at the diagnostic clinic, I would, we triage on a bench, but it was really awesome how it is, and I'll answer the questions at the end. So it's really important how on the bench, you would see it on the bench, but there's a window right in front of it, which was great because we would always hold up the leaf into the window. And you see these necrotic areas, I could see these, but all this water soaking greasy spots are usually not noticeable unless you hold them up to the light. So this is something that is really characteristic of bacteria. There are certain other things, some fungi can cause some water soaking, so it's not always a hard, fast rule. But almost always when you see this like windowing, greasy appearance, it's water. It's bacteria. And I always find that really cool. And again, I think it's really fun to show people how how the symptoms change depending on if you have a light shining through it or not. Here's an example of a strawberry leaf. And again, you have these advanced necrotic stages. And one other thing about bacteria is they tend to, they can really they have a hard time jumping veins. So they can't really go over veins. So a lot of times their spots are really angular. So if you can see, let's see, good spot. Like if you can see right here, it just looks like a perfect box almost because they can't really get past those veins really easily. But again, these necrotic lesions you can see if you're just looking at it on a flat background. But the second you put a light to the back of it, you can start to see all this extra little windowing, greasy looking spots, which you couldn't see before. And these are the early stages of the infection. So that's just a cool thing that bacteria do. And then I will answer the questions here. So there's two questions. Is there specific treatment for individual fungi? So as we progress, we get a lot of more specific fungicides. So a lot of fungicides, there are some that are broad spectrum that can control many different things. So they can control even some oomycetes. And then we also have more specific fungicides that are specifically targeted to certain groups of fungi. So there are fungicides that target oomycetes in particular. Methoxynium, I think, is one of them. So there are ones that are very specific. Um, so most of the ones that are, for usual homeowners are pretty broad spectrum, but they do get more specific. And then oomycetes can be very common pathogens. If you've ever heard of Pythium or Phytophthora or downy mildew, I've mentioned downy mildew a couple times, the one with the grayish um, fluffy growth on the bottom of the leaves, those are all oomycetes. So they're not as common as true fungi, but they are pretty common. Um, yes, fungi do cross veins every now and again. Some fungi will have that angular lesions, kind of like bacteria. So some fungi don't cross veins as well as others. But in general, fungi will cross the veins pretty easily. Um, Maybe not a mid vein, and that's why they have really circular spots. So bacteria have more angular spots, fungi will tend to have more circular spots. Okay, so now we're going to get into a phytoplasma. You may have heard about phytoplasmas before. These are a group of bacteria. So they're kind of usually referred to as a different entity, but they are inside the bacteria grouping. And basically they lack a cell wall. So they still have the cell membrane, so they're kind of just a cell, but they're very fragile. They cannot survive in the external environment. They can only survive inside their plant host or inside their insect vector. Um, so it's like a more weaker, more susceptible bacteria. So no cell wall. They are obligate plant pathogens. So again, they can't survive outside of their host or their vector. Almost always, they will live in the phloem of the plant, so that the plant's nutrient system, so they are interrupting the nutrients of the plant. So they cause these big symptoms, and a lot of times they cause death of the plant because they're interrupting its nutrient flow. And most often, they are transmitted by vectors. And mainly sap-sucking insects like leafhoppers and psyllids. And this is understandable because sap-sucking insects, they feed on the phloem. So it's a really easy way for the bacteria, the phytoplasmas, to get straight from the phloem to the insect vector, because again, they cannot survive outside for any amount of time. So there are quite a few common phytoplasma diseases you may have heard of before. Citrus greening, or HLB, also called Hualongbing. So it is, if you guys know anything about the citrus industry in Florida, it's kind of been wiped out by citrus greening, and it's a really big push to kind of figure out ways to manage it as well. But that is caused by a phytoplasma. And it's transmitted by Asian citrus psyllid. Lethal bronzing of palms is also very, very common, and it's causing decline in a lot of palms in Florida. That is caused by a Phytoplasma palmae. And then aster yellows. This is on aster plants. It's also caused by phytoplasma. I think I have a picture of it somewhere in the presentation, but it causes really weird symptoms where it just causes proliferation of the bud. So rather than like a flower, it's just like really weird growth. And then if you notice, all of these terms have candidatus in it. So this is another fun fact. So with phytoplasmas, similar to bacteria or similar to biotrophs that we discussed, how long can phytoplasmas survive in the vector? That's a good question. I don't know, and I think it probably depends on the vector. I would have to look into that, but that is really interesting. A lot of times it survives long enough for them to, jump from plant to plant, usually. But I don't know if it's like indefinite, if they can survive forever. I don't think they can reproduce in the vector, so that might mean eventually they will kind of fizzle out. But that's an interesting question. Okay, so candidatus. So that is a term, again, because these can only survive in the host or the vector, they cannot be cultured independently inside a lab. So we cannot, culture them on the sugary potato jello that we like to do. And as a result, we cannot 100% in terms of science meet the criteria for naming these. So we like to take out, we have a symptomatic plant, we isolate some pathogen, we make sure it's only that pathogen, then we inoculate it back into the plant, and then we see those symptoms of the disease, and now we know for 100% certainty this pathogen causes this disease. Even though we have all the genetic data to kind of prove that this phytoplasma causes lethal bronzing, we can't culture it out, so it has this candidatus in front of it. So that just means unculturable. So we don't have 100% proof without the confounding factors that this is the causal agent, but we pretty much know. So that's just a fun fact. If you ever are looking at something, you see candidatus, something or another. So this is the genus, this is the species. Candidatus just means it's a phytoplasma or a bacteria that cannot be cultured out or isolated. And that makes it really hard for science. That's one of the big issues when it comes to finding a control for citrus greening is it's really hard to, reproduce and to culture out the pathogens so that you can study it. All right, so those are phytoplasmas. Again, something that you will probably come in contact with sometime, and it's good to know what they are. All right, so we're going to move into viruses. Any questions? All right, so if there are questions, I can answer them. So here are two terms that we will see often referred to with viruses, and these can be very confusing. And I will say just off the bat, when in doubt, just say mosaic. So you'll hear mosaic more frequently. Technically, they have different definitions. Mosaic is when you have intermingled patches of green and yellow on the leaves, and they tend to have distinct boundaries. So if you see over here, this is really distinctive mosaic pattern. You have the chlorotic or the yellow areas with really distinct boundaries with the darker green areas. Mottle symptoms are more diffuse or irregular patches of light and dark areas. So over here, you can see that there's something funky going on with the leaves for sure. If it's something funky, it's likely a virus. And you see the yellowing or the light green, and then you have the dark green, but there's not like a really distinct boundary. So that can be more of a mottle symptom. The issue that we have is, as you can probably imagine, it's usually not always this perfect. So there's a lot of in-between zones where it's like, it's hard to really tell if that's a mosaic or a mottle. Also, a lot of viruses are named mosaic. So we have tobacco mosaic virus. But tobacco mosaic virus infects a lot of different plants. So on tobacco, maybe it causes mosaic symptoms. Maybe on tomato, it causes mottle symptoms. The symptoms are different depending on the host. So it's confusing. So in general, just know that there are these two terms. No one's gonna ever really like test you, be like, oh, that was not a mosaic, that's a mottle. It's confusing, but it's important to know that these are commonly referred to when it comes to viruses. All right, so we also have ring spots. Ring spots are common for viruses. And here I have circular chlorosis with green centers. And by green, I just mean the color of the actual tissue. One thing I learned that with fungi or bacteria, if you have a spot, the center will always be necrotic if the outside is necrotic. With viruses, you occasionally have this phenomenon where the outside is funky, but the inside looks like normal tissue. So for this potato, the inside has pretty normal tissue, and it's only the outside that is something is wrong. And so for this is a really confusing example because if you look at it, it almost looks like someone just stuck their thumbprint in there. But this is actually a virus. It's potato virus Y. And again, viruses have those ring spots sometimes. So this is an example on blueberry. And it's a little bit harder of an example because, if you look at it, potentially it could have been a bacteria or fungi, but it was tested to have a virus. And then this one's a real tricky one because if you look at this, that looks just like a fungal leaf spot. And maybe some fungi has come in afterwards. You have other stuff that looks a little bit more viral-like. So again, the center is yellow and it's chlorotic ring. It's probably here too. But this is tomato spotted wilt virus, which is one of the few viruses that sometimes also makes necrotic lesions. So again, with everything, it can be confusing and it's not always really understandable. But when you have these ring spots, this perfect circular areas that are not really necrotic, it's probably a virus. All right, so are there any questions with the pretty much the terms that are usually related to certain pathogen groups? If not, now we're going to move into these are general symptoms that the plants will display where you cannot really pinpoint it to a specific pathogen group. So if a plant has this symptom, it's not like, oh, that's most likely a fungi or, oh, that's most likely bacterial leaf spot. These can be caused by different sources, really. So first we have canker. So the definition of a canker is a localized dead necrotic area, often sunken lesions on the stem or branch. So this is a good example of a canker. It's a small area, so it's not, the whole plant. It's usually a small section of the plant. It's sunken and it's necrotic. It's dead. This is also a canker, and I put this here because this is citrus canker. It's a pretty common disease here in Florida. And it's actually not sunken, it's raised. So again, even with these definitions, they're not hard, fast rules, and scientists don't always follow the nomenclature that well either. So the disease name is actually citrus canker. So it is a canker, but it's more raised rather than traditionally sunken. But typically, if it's just one localized area that's necrotic, it might be referred to as a canker. And then this is bacteria, and then this is fungi. So it's one way to say that you don't always know. I would say more often cankers are caused by fungi, but you can't always be certain. Chlorosis, again, this is chlorosis. We all know chlorosis is just kind of yellowing due to a lack of chlorophyll. Sometimes it's caused by nutrient deficiency like this. It can be caused by insects sucking out the sap. This is a fungi pathogen. It's caused chlorosis. This is a bacteria. But one other thing that you will sometimes see is a chlorotic halo. And this is just when you have a necrotic lesion that is surrounded by a yellow halo. So typically it's kind of where the pathogen is expanding to. So here it's a fungi. You have this necrotic lesion, and then a lot of times it's creating a toxin that's kind of creating the outside tissue to start decaying before the fungi really reaches there. So that's kind of a chlorotic halo. And these ones are pretty large chlorotic halos. A lot of times you have something like this, where you have, again, just a small necrotic area and then just that little halo surrounding it. I would say most of the time, chlorotic halos are associated with bacteria, but that is not always the case. Again, this is a fungi and it has a chlorotic halo. So that's just another term you may have heard at some point in time. All right, so now let's go into dieback. So dieback is basically progressive death of shoots, branches, or roots, starting from the tip primarily. A lot of times we refer to dieback with shrubby plants. So if you have one whole branch that's dead, that's usually dieback. So in this case, it's a sweet viburnum. Most of the leaves are green. You have this one branch that has died. And sometimes that is, it could be due to, mechanical injury. It could be due to fungi infecting, the base of the branch, or it can be a root related issue, a crown related issue. So the crown is the part of the plant that's like right at the soil level, not with trees. Sometimes trees, we refer to crown as the top of the plant, but also we refer to as crown as where the stem and the roots kind of meet. And then something like here. So you can see this is clearly starting to die at the tip. And again, so it's really just the death of one branch or one area. And then blight. So blight is something that it can be a little subjective. And a lot of times it's referred to as something that happens really fast. So this can be something for, like, as we discussed, the hemibiotrophs. Where they have like a biotrophic stage, so they're feeding on the living material, and then they switch to a necrotrophic phase. And once they switch to that necrotrophic phase, they just rapidly kill the plant. And this can be described as a blight. So this tends to be something that's just pretty fast. And it's rapid killing of leaves, flowers, stems, or just any part of the plant, really. Typically above ground. So this is southern blight on begonia. Again, it's like one day it's fine, the next day, boom, it's melted out. And as a fun fact, you see those little seeds, those specks that are kind of around here. Those are sclerotia. So they are the hardened fungal hyphae that will live in the soil for ages. Um, so if we see sclerotia, that's not a good sign. We need to switch to a non-host fast. Or in this case, it's potting mix, so you can kind of just get rid of it. And this is Rhizoctonia blight on her hydrangea. Again, it might just start as a little lesion, and then all of a sudden it's starting to take out this whole area. Rot. So rot is just decay of plant tissue. Typically, it's from a pathogen that creates enzymatic activity. A lot of times it's associated with bacteria, so this is bacteria. And it's usually leaves the really soft, mushy tissue. But there are specific rots that are called like dry rots, which is dry, crumbly tissue. So the most time when we think about rots, we think about really wet, disgusting stuff. But there are some rots that are dry. And so this one was actually inoculated. So you can see the little puncture wounds. One way that if you're in a lab and you want to see if what you've cultured out is actually causing the disease, is you inoculate it into a healthy plant. So this case, they just shot some bacteria in here, and as you can see, it's starting to take over and kill this poor little orchid. And then this is actually a fungi-related rot. So again, rot is a lot of times associated with bacteria, but not always. Sometimes it's associated with other things. And again, this is just caused by fusarium rot. It's just a cutting that did not take, really. Galls. So galls is I love galls. It's like a special place in my heart because there's so much we don't know about galls. And that fascinates me. I love not knowing things sometimes. So galls are broadly defined as swelling or overgrowth caused by infection. So this can be caused by a lot of things: insects, mites, fungal, bacteria, nematodes, they can all cause galls. And it's just some part of the tissue gets swollen. And an important thing to note is that this is plant tissue. So the pathogen interacts with the physiology of the plant and causes the plant to produce these. So it's not, an example here, this is on Mexican petunia. I see this everywhere. And I like to put this here because when people show this, they almost always think that it is fungi because it looks like a mildew. It's like whitish growth on top of the leaf. But what it really is actually, it's a mite that's causing this, an Aeronose mite, that causes the plant to just produce a bunch of hairs. So all those trichomes, which are the hairs of the plant, it just produces a bunch of them and it creates like this little forest. And the mite really likes living in that little forest. It's humid, it's, really good. It's protected from the environment, from predators. Like it's a really good house for the mite. So this is called an Aeronose gall. So it's a bit of an unusual gall because it's kind of just a lot of hairs. But yes, if you ever see Mexican petunia, look at them for whitish growth and just marvel in the fact that it is a tiny, tiny microscopic mite that created those symptoms. So here's an example of a fungus that can cause galling. So this is on holly. It's Sphaeropsis gall. Sphaeropsis is a really hard fungi name to say, and it took me forever to learn to pronounce it. It's pretty common on woody ornamentals. It's typically caused by pruning, just not sanitized pruning gear, pretty much. So if you just go from one thing to another, you might end up spreading this fungus. It also causes galling. It can sometimes cause witch's brooming symptoms, which I'll have on the next slide as well. And again, it's causing the plant to produce these growths. This is an example of a bacteria. It's Pseudomonas gall on Loropetalum. This can be pretty common in nurseries, less common in the landscape. But as you can see, these kind of like little growths over here, it's a bacteria that's actually causing the gall. And then here are nematodes again. Nematodes are kind of their unique group. So you see these little bumps all here on these roots. This one is just swollen. It's big. You have a big one here. That's the nematode. They burrow into the roots. They cause the roots to swell, and the swollen roots produces more food for the nematodes pretty much. And so again, there's another group that also causes galling. So a lot of different organisms will cause galling symptoms. And then witch's brooming. So this is another really unique symptom, and this one is really, really fascinating because again, there's not a lot we know about it. But it's also swelling or overgrowth caused by infection. So it's similar to a gall, and they can be a little confused. In general, witch's brooming happens on the end of a branch, or they tend to be the bud is proliferating a lot. So instead of having one branch come out of a point, you have 100. So that's kind of what's going on here. Rather than one bud being produced, you have like 100 buds. This is also caused by an aerophyte mite, so the same mite that cause a different species, but the same type of mite that causes this causes these, and that specific type of mite causes a lot of these really unique symptoms. And then this is also a phytoplasma. So again, those bacteria that lack a cell wall, which are phytoplasma. This is Astra yellows. Again, instead of having a beautiful flower or a different bud, you have just 100 of different buds. Really fascinating. Also, herbicide injury can sometimes cause witch's broom-like symptoms as well. And then also another important thing to mention with witch's brooming and galls is a lot of times the reason why these pathogens or pests produce these is because they're really good homes for them, and also they can produce extra food for them. But the thing is, sometimes other insects or other pathogens will come in and be like, hey, this is a really nice home. I want to live here. So if you see a weird symptom like this and you look at it and you see a bunch of insects, you're like, oh man, these insects caused this gall. Well, that's not always the case, or a lot of times it's they moved in afterwards. So it's really hard to prove causality, which is one of the reasons why there's a lot that we don't know about witch's brooming and galling symptoms. So yeah, proving causality again is really, really challenging. There's environmental factors that come into play. Again, it's also the pathogen or insect is it's actually like altering the plant, and it's really hard to replicate, really hard to study. It also, most of the time, it affects a lot of ornamental crops. And so there's not a lot of research around ornamental plantings, so there's less research. And so there's just a really big gaping hole of understanding when it comes to galling and witch's brooming symptoms. So just a fascinating area, but I will caution you, if you see insects inside a gall or a witch's broom, do not automatically assume that they caused it, because sometimes they just see this and like, that's a nice house and I want to live there. All right, so now I'll just stop again for questions. After this, we are pretty much done. I just have some practice afterwards, I believe. All right, so you can type your questions in. I think we will, oh, I have one more thing, so I'll talk about some diagnostic tools. This is something else that I can expand upon in the future if there's a desire for that. All right, so for diagnostic tools, the first thing that's always important is get in close. Sometimes if you look at a leaf spot, it just looks like a necrotic area on a leaf. You take a little closer look, you can see fuzzy stuff on it. And so that might indicate that maybe it's a fungus. So obviously you need kind of some training to get to very specific identifications. But if you just get closer, a lot of times you can really narrow it down. So there are lots of field guides and apps. iNaturalist is wonderful, and I feel like the more people use it, the better it gets. It's really good with insects. It is lagging behind when it comes to diseases and other stuff like that, because again, the symptoms on a plant can usually be referred to many different causal agents for that. So again, these field guides and apps can definitely help you narrow down the search. But I would caution you when it comes to diseases, trusting everything that's on those apps, because it is really hard to tell without sending it to a lab and actually culturing it out and having a specialist, look at the spores and stuff like that. We also obviously have a bunch of UF resources. So a lot of EDIS publications. You have us extension agents. Again, I'm here as a resource. I love talking about diseases, and I miss kind of the detective work of getting a sample in and figuring out what's going on with it. So I'm always here to talk about diseases for sure. Unfortunately, most of what I feel like master gardeners deal with is a lot of homeowner and just ornamental plants, and that's really lagging behind with a lot of research. So that's kind of unfortunate, but also kind of exciting. Sometimes you look at it, there's new things always being discovered because there's not a lot of emphasis being put into ornamental production. Um, and then finally, we have diagnostic labs. So this is the lab in Gainesville where I worked at for, almost 4 years while I was getting my doctoral degree. Um, if you, and honestly, sometimes we would come back being like, we don't know, it's really hard without the full story. But if you want to get the best guess, send it to the lab. It's $40. They will send you a result within a week. Um, and they're good. They can use that data to help guide efforts in the future. And we have labs closer to us. So there's one at the Gulf Coast Research and Education Center. And then Kathy asked, are there simple cultures we can perform in our lab? I am looking into that. So there are definitely things we can do in our lab. So one thing that I can easily do in our lab, and I would love to if we one day have like a microscope training, I think we might do that sometime in the future. One thing that we do a lot is a tape mount. And so basically you take a little glass slip and you take some tape. So you take one of these. So one of the glass slips that you put under a microscope, you take a piece of tape, you tape it onto the necrotic area, you take it off, you put water on it, and then you can see the spores without having to really culture them. So if it's something that sporulates on the plant, it's one way to identify the spores without having to culture it. So that's something that is really important, and it's really easy for us to do. The problem is sometimes not everything sporulates on the plants. We can do moist chambers for sure. So a lot of fungi just need a little bit of moisture and they will just blossom. So if you just put them in a baggie with wet cotton or like wet paper towel, leave it for a day, a couple days, boom, you have fungi galore. Um, Also, a lot of there are simple like Petri plates, so simple PDA plates, potato, dextrose, agar. Those are pretty easy to make. And again, you can, if you're in a lab, you have, incubators that have specific, 12 hours of light, 12 hours of dark, specific temperature, specific humidity, but you can easily do a base level cultures without that. So there are definitely very simple things that we can do in our lab. The easiest one is to just moist chamber it, and then you can just look at what you can see under the dissecting scope, and then also do a simple tape mount. And that's really easy to do. Other than that, GC Rec is not very far. It's like 40-minute drive from here. I can easily send it there, and I could even just plate it out there. So there's lots of options. But yeah, maybe in the future I can work out a little bit more of like an actual lab here. Um, all right. So I'm going to go into the practice round, and then we can end with just a bunch of questions if you have any questions. So this is just, I'll have a picture basically, and if you just throw out some words that you would use to describe this picture, and then I'll kind of describe what it actually is and just a little bit of practice. So when you see this, it's a branch with this big growth. What would you use to describe it? We already have some galls. It's really great because yeah, and again, this is a plant tissue. This is a gall. And again, we don't really know off of just observation what is causing it. We can have some clues, but this is actually caused by bacteria. So it's caused by bacteria crown gall, which is Agrobacterium tumefaciens. And then I have a fun fact about Agrobacterium. So this was the first agent that was really used in order to genetically modify organisms. And I have a video. Again, I have the PDF version of this I sent to Kathy, so you guys will get it. So if you want to see like the actual video of how it happened, but basically the bacteria has, it will take some of its DNA and it will insert it into the plant's DNA. And the DNA that it inserts causes the plant to grow. So it changes the hormones, so the plant grows really big, and also causes the plant to produce a specific food that only that bacteria can eat, so feed on. So it's a really awesome way for the bacteria to really optimize its growth. But what scientists do is they take out that DNA and they insert whatever their own DNA, and so that they can use the bacteria's mechanism to insert the DNA into the plant, but they have different genes. So that was the first way that we really did genetically modifying organisms. And it's still used widely today in science. We have other methods like CRISPR, but this was like one of the first methods, and it's still used today. So, fun fact. All right, so this one we've used again, but I just want to also add this one here. How would you describe it? What are some words that you would use? It can be a little bit of a tricky one, which is why I added it. Canker necrotic. So yes, it is necrotic for sure. You see that dark halo. That's a really good one because again, you can see that chlorotic halo. Target, yeah, it's kind of has a target spot too. Especially over here, that does kind of look like a target spot, if I'm being honest. So yes, this is, canker, citrus canker. So it's a canker. It also has chlorotic halo. It has some necrotic lesions. Um, and again, cankers tend to be sunken, but in this case, they're kind of raised. But all those words would be a really great way to describe this if you were sending a picture or an email to someone. And then the fun fact, if you don't know about citrus canker, is it was introduced into Florida 3 times. It was successfully eradicated twice. And then the last effort to eradicate it, it ultimately failed because there was a couple of hurricanes during that season that spread it really far. And also people really were opposed to the USDA coming in and cutting down their backyard citrus trees because they had canker in them. So a lot of reasons, but ultimately it was failed to be eradicated. So now citrus canker is all over Florida. But ultimately it's not that big of a deal because shortly after that, citrus greening came in and that kind of um, made canker look not as bad in comparison. All right, so this one might be a little tricky. So how would you describe this? This is Bermuda grass. And so what I'm specifically looking for is like this little area. How does this one differ from like over here? Like what words would you use to describe? And this is a closer image of the actual, basically this. Fungal, chlorotic. So yeah, it's definitely yellower. So this is definitely chlorotic as opposed to over here. Stunted. Stunting is really good. Yeah, this is much smaller than over here. Something's going on with that for sure. All right, so the other word that I was looking for is witches brooming. So again, the witches brooming is when you have, instead of just one bud or one branch or one leaf blade coming out, you have a bunch of them. There's a whole bunch of like new growth that's just competing and it's not coming out how it normally would be. So this is actually caused by a mite. Again, those aerophyte mites, which cause a lot of times witches brooming or galling symptoms. Um, so these are the actual mite itself. They are very small. You will almost never see them. Even I've tried many times to see the mite inside the Mexican petunia galls, and like every 5 samples I get, I will actually find a mite. It's like nearly impossible. They're just so good at hiding. They're so microscopic. They're so small. And they actually are really interesting. Here's a electron microscope scan of them, so really bloomed up. They're interesting because they only have 4 legs. So you see 2 on each side, whereas most mites have 8 legs. So they're a very unique group of mites. One of my favorites. They look like little sausages. They create really funky symptoms and they are really hard to find. So they are a bugger if you're trying to identify something, but that makes them fascinating as well. So yeah, they cause witches brooming symptoms, stunted, chlorotic. It's just funky. One way to describe it's just kind of funky. Um, all right, so let's do this one now. How would you describe this? So it's obviously strawberry. This is 2 different stages. This is a nice green strawberry. This is a almost ready to eat strawberry. Rot. I don't know if it would be rot necessarily. So this was one of the first terms, so it might be hard. What I was really thinking about is something this may be good. Mildew. So this is called by powdery mildew. So again, mildew is something that has whitish growth on the surface. And this is all whitish growth. And I like to include this one because I think this is really pretty when you look at this under a microscope. So this is, and if you see this, this is actually the spores of the fungus and some of the hyphae mycelium. So this is a sign. So if I look at this white stuff is a sign, not a symptom. Um, Yes, Nancy, you said that. Yes, are these signs? Yes, they are signs. Wonderful. I love that. So yeah, these are signs, not symptoms, and that white stuff you see is the actual fungus. And then one thing about powdery mildew on strawberries is a lot of times they coalesce around the seeds because that's a really easy place for them to kind of escape. So they infect on the inside and then they kind of just escape around the seeds and they kind of push the seeds out as they're trying to escape. But yeah, I think it looks really pretty and these are all the spores. Um, yeah, so I think this is the last one. So this one has a bunch of our words. So how would you describe this? Target spot. Yes, you can see those concentric circles. It's very circular. Again, fungi tend to have more circular spots. So there's also this hole here. What is that called when it's the hole kind of falls out? Sunken. They are a little sunken, maybe. They're definitely necrotic for sure. I'm looking for a shot hole. So if you get that hole, it's like a shot hole. You put a shotgun or a BB gun through it. And we also have, what is this yellow stuff on the outside? Not crossing veins. That is true. So again, fungi, um, they grow circularly, and they tend to have a hard time crossing these types of veins. But all of the little veins that are in, oh wait, I'm pointing at the wrong thing. So fungi, they have a harder time crossing the main vein. So something like this, they will have a hard time crossing. But there's also veins throughout here and here that fungi can cross over that bacteria might have a little bit of a harder time crossing over. And so the last thing was just a chlorotic halo. So their target spot, again, concentric rings. We have chlorotic halos, that corroses all around it. A shot hole is that looks like the centers are falling out. So this is target spot on tobacco caused by Rhizoctonia solani. And here is, here's an example of, this is the asexual stage, Rhizoctonia solani. The teleomorph, which is technically the sexual stage, is the Nanophorus cucumeris. So again, two names. So some fungi will have two names. And then this one did not follow the standardized rule because technically we're supposed to call it by its sexual name, but no one knows what Thanatophorus is, and a lot of people know, if you're a plant pathologist, what Rhizoctonia is. So again, another callback to fungi are confusing and they have two names most of the time. All right, so I think that's all the practice I have. We can always do more practice in future lessons. And here is just a comparison table. So this is something just for you to reference. Again, you'll have the PDF if you ever want to. This is a broad generalization. So do not use this as the Bible, but this is really good to kind of narrow down your search if you're looking for a fungi, a bacteria, or a virus, or an abiotic cause for whatever symptoms you're seeing. So for example, if you have water soaking, a lot of times it's bacteria. Occasionally it's fungi. Occasionally some abiotic stuff. So if you see water soaking, it's probably bacteria. Rotting smell, most of the time it's bacteria if it's rotting. Sometimes it's fungi. Necrosis, a lot of these have necrosis except for viruses. They don't typically, but there are some that do. And then bacterial ooze, this is just saying that bacteria rarely cause ooze, so most bacterial diseases will not cause ooze, but if there is some bacterial ooze, obviously it's going to be a bacterial disease. So again, if you look through this chart and you see these symptoms, you can kind of get a narrower view of what you might be dealing with. But it's not going to be, there's always exceptions to the rule. All right, so I know I'm way over time and I feel kind of bad. I practice it and I guess I usually talk faster when I am doing things in person versus practicing, but Here's a survey. So as I discussed, this is one of my first big presentations that I kind of developed myself. Learning a subject is one thing. Learning how to teach a subject is a very different concept, and it's something that I'm still figuring out. So it would really help me to kind of cater my future programs if you just kind of help me figure out what's the best way, what I got across well, what didn't get across well. And then also there's a spot in the survey for if there's anything you want in the future. I can definitely dive into many other topics. And we'll hopefully have some hands-on topics at some point because those are always fun. So are there any last questions or anything else? I am here to answer anything. No more questions. All right, and just let me know if the survey doesn't work. You don't have to fill it out now, obviously, and then obviously here's my email. So uh, more than happy to have questions and concerns. I love pictures and I love samples, so I'm really happy to look at things under a microscope for anyone. So thank you so much. And I'll just stay on here. You guys can leave if you want, or if you have any more questions, um, then I will stay on if you want to ask them just more individually. And thank
Thank
you
you
all.
so much, Caitlin. That was very, very informative. And for a difficult subject, it was surprisingly clear the way you presented it. So thanks.
That's great because I've been to a couple of these presentations and it's really hard. Some experts are they're not good at teaching these in a way. And so I look at the other participants and I can see their eyes glazing over. And so I don't want to be like that. So I would love to really hone this so I can hopefully become better at explaining it in a way. Because this is my favorite subject. So I want people to really love it as much as I love it. So yeah, I want them to be able to actually understand something that is so confusing.
Yes, I can tell you love it. And that really, yeah, makes it that much better. So good job.
Mhm.
And I hope everybody will stay tuned for, we hope to do these once a month and take deeper dives into diseases and even some practical hands-on, actually looking at things and diagnosing problems. So
Also, I think it would be cool if you guys see examples of kind of keeping them and you can show them to me. And I think it could be cool to just have like little reviews. So in my old lab, we used to have this section in the lab where it's just fun stuff. And so that everyone can kind of experience something if we weren't in the lab at the time. It's just something that we share with everyone if it's something that we're experiencing, if something comes into the clinic, you kind of just broadly share it with everyone so we all kind of learn from what's going on.
that's a great idea. We often do that with insects and but
Yeah.
yeah, diseases are really one if we have a positive identification too, that really helps.
Yeah. And I have a pretty close connection with the GC lab because I worked there for a semester. So I'm always willing to go over there and um, either I was like, if you guys are busy, I'll just do it myself. But they have the equipment. So it's a little harder to culture things out here.
Mhm.
Um, but we can also just send off them for them to do the diagnostic.
Great. And there's a question, will I distribute the presentation? Yes, I will. I have it and I can send it out to everybody. And this was recorded, so I'll send the link as well.
Can you send it to me as well?
Yes.
Right.
All right. Are there any other questions, comments before we close out? Okay. Well, thank you, Caitlin.
Thank you.
Hope you're improving every day, but
Yes,
sounding great.
I am so happy I made it through this fine.
Yeah.
So my second cold this season, so it's been kind of really hitting me.
Mm.
Uh, I think it's because I
Well,
broke my wrist and my body just cannot. It's building
it,
bone and it cannot fight off any viruses. So
yes. Well,
I
at least you're healed from that, it looks like.
yeah, oh yeah, no brace. I actually was discharged from physical therapy yesterday, so uh definitely doing better.
Good. All right.
All
Bye,
right,
everybody.
I will leave as well. Bye. Thank you guys.
Mhm.